So I would love to talk to you, thank you for Steffi for organizing this, about stem cell derived organoids. Is it the new frontier in medicine? I have this up because this is what you do in the States. One of the reasons why I came back is actually shown on this slide over here. I was able to be picked as the executive director for this new center at the TUM, the Center for Organoid Systems. And as you can see on the right side, we have a collection of really the critical organs that we are trying to rebuild from stem cells. The brain, the heart, the liver, the pancreas. But we're combining it with absolutely cutting-edge technologies, as you can see on the left. And I think this is one of the strengths that we have at the university, at the technical university in Munich. And what we'd love to do is essentially shown in the bottom. We would like to explore innovative organoid technologies for the analysis of human diseases, but then, and this is important, for the translation and the development of novel therapies. So I'd like to start out, I was surprised how many people already know about organoids, so maybe I don't have to show this, but I usually frame it like this. All of us, hopefully you can recognize yourself on this slide here, we of course are made of a number of different organs. It's obvious, right? But all of these organs consist of cells. Individual cells. In each of our bodies, we have approximately 250, and still growing, different cell types. But what I find really amazing is that all of us came from one cell, the fertilized egg. And so when we bring this back in this direction, when we are a fetus or an embryo, we are growing from the left to the right, and that is the idea of organoids, doing the same thing. So the analogy I often use is just of a Lego block. They come in all kinds of sizes, different forms, and even colors. And as we've heard this morning from our minister president, who seems to be my age, if you really put these things together, you can build the Millennium Falcon, like in Star Wars. And that's the idea. How can we take these cells and build organs out of this? Very complex structures. And the cell that we need for that, of course, is a cell that's equivalent to the fertilized egg. And do we have those? The answer is yes. We actually have two different ones of those. Embryonic stem cells that unfortunately have to be derived by destroying an embryo, and very, very few people do this. What people are now using, most of us, are induced pluripotent stem cells, cells that we can take from your skin right now or from your blood, modify them, put them in culture, and then within culture, they can stay for weeks, months, years, actually decades, and what they maintain is their ability to differentiate into a number of different cell types. Blood cells, muscle cells, but also more complex organs like brain organoids, not a complete brain, heart, and what we are interested in is the beta cell. So the idea, of course, therefore, is we can rebuild these cells, we can use them for cell therapy, and we can do this for neurons, Parkinson's disease, or myocytes for cardiomyopathies, or, which I will tell you in a second, for stem cell-derived beta cells. I find this amazing, and I just show this slide. It's from one of my colleagues, Alessandra Moretti, who works on the heart. The heart is a really complex organ. It not only consists of a number of different atriums and ventricles and all this kind of connective tissue, but it also, within the cells itself, it's not just one cell type, it's a multiple of cell types. And only when you have that do you get this beating heart. 60 times a minute, 3,600 times an hour, 86,000 times a day, every day of your life. And what they have been able to do is miniaturize this and put this, and this is in cell culture, and there you have a structure that has similar composition of what we have in the heart. It's way smaller, it's not a full heart, of course, but it starts to beat. So this is, again, is a way for how we address these things and try to rebuild these things. We work on something else. And I often show this picture to just orient myself and remind myself, but also you, why we're doing this. This is an old picture. It's a young boy who's being held by his mother, and this young boy is about to die. This picture is 104 years old. So in a way, this young boy was fortunate because when he was diagnosed with diabetes, type 1 diabetes, insulin became available. Insulin is a miracle drug, has saved countless lives over the last century. But it is not a cure. What does insulin do? If you have on the left here a healthy person within what we call the Langerhans Islands. It was a German pathologist who described it first, or islets of Langerhans. We have these blue molecules, insulin, being secreted when the sugar levels goes up in our blood. They go in the bloodstream, they go into circulation, they go in the peripheral tissue, and then in the peripheral tissue, fat, liver, and others, muscle, the glucose is taken up, so that our sugar levels are in a very narrow range when you're healthy. If you have type 1, you have a destruction of the beta cells because the immune system destroys them. If you develop type 2 over time, you still have beta cells, but they don't function as well as they should. But the outcome is the same. At the end, you're just not producing enough insulin to regulate the glucose levels that you need. And in that case, if this is the case, you have complications, cardiovascular, kidney, and so on. And those complications can be life-threatening, and in many cases they are. So if insulin is not a cure, is there a cure for diabetes? And the answer to this is yes as well. Many, many years ago, what people figured out is that, unfortunately, someone dies, we can take out the pancreas, and we can isolate the islets, and we put them back into a patient with diabetes, and that patient then, for all intents and purposes, is cured of diabetes. I show you real-time data here. This is actually from UCSF. This is a patient who did everything right, was on an insulin pump, but it couldn't regulate the glucose levels. That person had an islet transplantation. As you can see, the glucose levels dropped down in this narrow range, which, by all intents and purposes, is the same range that we have. It's the normal range. So why are we not doing this? Why are we not just giving everyone a cadaveric islet transplantation? The answer is we just don't have enough of these cells. There's very, very few in Europe and the United States, worldwide, that are available for this. And that's why we, as well as several other groups around the world, approximately 20 years ago, a long time ago, we asked the question, if we can't use the cadaveric islet transplantation, is there a different way to generate the cells we need? And what we decided to do is take these pluripotent stem cells that you see here on the left that I talked to you about, embryonic or induced pluripotent stem cells, and follow the differentiation stages that they would undergo when they are in a fetus. All of you, everyone in this room, has done this. And we essentially take this information that we've learned from developmental biology to generate these functional beta cells. And it works. It already is in clinical trials. There are patients out there that have been insulin independent. They don't have to inject themselves anymore with insulin for more than a year, and those are approaching now 20 or so. And therefore, what I would say is that what I would call the SC, stem cell-derived islands of the current generation, they provide glycemic control. It's very good already. But they have metabolic and functional defects. And therefore, what we are doing right now, as well as others, we're trying to understand how we can modify them further. One way of modification is to modify the immune system so that the immune system does not destroy them. Because even if you transplant them, those patients have to be in immunosuppressions. But where I really want to go is here. I want to generate the future generation of stem cells, and that's what we're doing in the lab right now. Cells that have enhanced longevity, they last longer. Cells that have optimized metabolism, and they are self-healing. We want to generate smart cells. And I'll just give you one example of what I mean by this. Just one snippet. If we transplant cells in any patient, the first thing that happens is that these cells will have hypoxia. They're exposed to low levels of oxygen, and the beta cells, unfortunately, need high levels of oxygen because they're very metabolically active. So what we are building right now is cells that have a smart oxygen sensor, and as soon as the level of oxygen drops down, they drive the expression of a genetic circuit, and that blocks the cascade of hypoxia. And just to tell you how we're doing this, we are developing sensors that under normal oxygen or high levels of glucose, they're being degraded in the cell, but as soon as the level goes down, and only then, do they get activated, the sensor becomes stable, and it can activate the expression of genes. And this is an example of this. Under normal oxygen conditions, you don't see any expression. Under hypoxic conditions, these cells get active. So essentially what we're trying to build here are cells that can respond to stressors that they might have when they're sitting in someone's body. The second that we are doing is also has to do with transplantation. The company has started in San Francisco, where we can actually put nanoprobes, these are very, very tiny little molecules, gold particles, into each of these stem cell-derived beta cells. And what that allows us to do now is to go after transplantation, put a light beam on it, and get a signal out. So essentially, we can now follow in real time how the transplant behaves over time after transplantation. Essentially takes away the black box that we have. So what we've done, all of us, over 400 million years since we left the water and looked like this is how we are today. If we would have another X million years of evolution, we would certainly change. There's just no way around it. But what we would like to do with the beta cells is, I would pose that where we're here right now, we can get to the flying guy, not in X million years, but in the next 5 to 10 years. And now I'm going to say it, AI is going to help us do this. This is a reprieve on AI, by the way. I hope you figure this out. Okay, good. So I have a couple more minutes, I believe. Oh, this is going to be fun. I want to tell you a little bit about what else we do, because we also work on the dark side. The dark side is pancreatic cancers, and we're going to use the cells not for cell therapy, but for tumor modeling and drug discovery. The pancreatic cancer is a really bad disease. It is an exocrine tumor coming out of these acinar cells that produce enzymes that are now active because you have just had food, and the duct system that essentially transports these enzymes into the gut. Pancreatic cancer is one of the most dismal diagnoses in all of medicine. Actually, as we are right now, it's around 12 or 13% of patients that have a 5-year survival chance. It's at the very, very bottom. And in the United States, in 26, it has moved up to be the 3rd leading cause of cancer death already, and it's projected to become the 2nd highest. So we need to understand this, because most of the patients are diagnosed late. We need to go and identify markers for early diagnosis and vaccination. And what we decided to do is to build these duct cells because they can turn into pancreatic cancer. And so how do we do this? This was done by us, but also published by some others. We follow the same idea. We use the stem cells, we give them molecules that they would see during the development in the embryo, and we turn them into what we call duct spheres. They express the right markers. These are just colors for markers expressed in duct cells. And if you see a section through it, which is a little bit hard to see with the light, but you can see it's almost like a ball-like streak. But, of course, none of us has little balls in our pancreas. What we have are elaborate structures and networks of intercalated and interconnected ducts. And that, of course, is something that we need to rebuild to understand where the cancer is coming from. So we went back to the drawing board, we modified it. We're now using hydrogels, combinations thereof, and now we get into duct organoids. And this is shown over here. I hope you can see this. We can now build structures in light blue that are significantly changed. This is 0.3 millimeters, so several millimeters in length and in complexity. And if you go in and take a look at by a movie, I hope you can appreciate this, that this is a real tube that we now can build under just cell culture conditions, just in the Petri dish. So where are we going to go with this? And I'm out of time. It's blaringly red, so I'm going to go fast. In patients, 90% of the patients that have pancreatic cancer have a modification, a mutation in this one oncogene called KRAS. Mutation is called G12D. So from the normal duct to dysplasia to cancer, KRAS is the driving force of this. And now, again, our system allows us to modify this. It's very versatile. We have ways to activate KRAS. And if you do it in the normal duct, then over time you can see how the duct changes. And I just have an histological section here to just show you, and I think you immediately can see this. This is the inner part of the duct structure, and it's a one-cell layer epithelium, exactly how it is in our ducts. And if you activate KRAS, it looks like this. There's multiple layers of the epithelium that are in there, multiple cells are in there, cells are coming off. This is a mess, unfortunately, like the cancer works. We can look for markers. The markers that I show here, just the only thing you have to take away from this, everything that lights up, is very similar to what we have in pancreatic cancer. So, and this is one of the last data slides I have, how close are we to pancreatic cancer? what we can do now is called a single cell analysis. So we can go and just look at the duct cells that any one of us would have in our pancreas, and we can visualize it. And this is the population of cells. They are in blue. These are the normal duct cells that you have. Now, if you go in and look at just the cancer that you would have in a patient with pancreatic cancer, and you superimpose this in gray, you see there's a little bit of overlap, but not a lot. These cells have moved away from where they normally sit. These are malignant cells. They cause cancer. So where do we stand with our cells? Now we can look at our stem cell-derived cells that have this activated oncogene in them, and I hope you can see this, that there's a few that are still in the normal range. I go back and forth, you see where they are, but the vast majority has moved out of the normal range, and some of them sitting just on top of the cancer already. So we can move towards building cancer in just 4 weeks, or at least the early stages of this, under cell culture conditions. You can do a secretome analysis and find biomarkers. You see over time, everything is red, it gets increased. These are markers you find in pancreatic cancer. We are finding some markers that people have, as of yet, not found. And so what are we going to do with this? Again, we're very fortunate because at the TUM, I'm part of the Center for Smart Drug Design that Stefan has put together. And there are people who are working on AI, they're designing molecules, they have designer biologics, and we're using our system to do this. And this is the last thing I'm going to show you. We have modified this now. Instead of having this duct structures all over the place, we are putting them on chips, and now you see just one length of a structure, which is likely much easier to use for drug discovery. I'm done. I just want to tell you that we use, of course, gene editing, and here it is. We use AI as well. Not me, I don't understand it, but we have some fantastic people we're working with, like Fabian, for example, Fabian Theis. We are trying to optimize the system as I just described it to you, and I do believe that, yes, we can generate cells that are better than what we have in our bodies in a relative short period of time to improve cell therapy. And we're also working very hard on stopping this cancer before it becomes aggressive. Last page I have up is an article from the New York Times in 1903. It's published on October 9th. So there's 2 reasons why I show this. The first reason is it's the day after my grandmother was born. And the second one is probably the more pertinent one for this lecture. This person was a reporter who went to Langley and was looking at how people were designing flying machines. And he says, the ridiculous fiasco which attended the attempt at aerial navigation at the Langley flying machine was not unexpected, and then he goes on. And then at the end, in the conclusion, he says, this is never going to work. They're going to use mathematicians and mechanicians, and they're going to use 10,000 years, and they're going to use zillions of dollars, and we're not going to fly. And 8 weeks later, we are flying. This is where we are with stem cell therapies. This is where we are with the organoids, and with your help, as well as the help from the government and our funding sources, which I have down here, I think we can change people's lives. So thank you.